Welcome to a tutorial of the physiology and control of a muscle contraction. Before we begin, here are the 4 functions of muscle. Muscles allow us to produce movement, maintain posture, stabilize those joints, and generate heat, particularly whenever we utilize ATP during that contraction. This tutorial will cover the last of our goals for this unit. First, we'll begin by reviewing the organization of a muscle. The organization begins with the muscle, such as the brachioradialis. This is made up of bundles known as fascicles, and each fascicle is made up of our muscle cells or muscle fibers. Muscle fibers are made up of these myofibrils containing filaments known as actin and myosin. Wrapped around each bundle is connective tissue. We begin with the epimysium, which will cover the entire muscle bundle, followed by the perimysium, which surrounds each of the fascicles. And then around the muscle fiber itself, the cell, within the fascicle, we have this known as endomysium, which maintains all of the nerves, arteries, and veins, keeping it nice and close to the muscles. Surrounding each muscle fiber is its cell membrane known as the sarcolemma, which is a specialized plasma membrane that stores glycogen as well as myoglobin. Myoglobin is responsible for holding on to all of the cell's oxygen for cell respiration, which will be used within the mitochondria to produce the necessary ATP. Within the myofibrils contain the functioning unit of the muscular system known as sarcomeres, which contain overlapping filaments known as actin and myosin. The sarcomeres are segments all along the entire fiber. A muscle contraction occurs when the myosin and actin come together. The myosin will pull on the actin, bringing the two ends of the sarcomere closer together. As you can see on the right hand side, the myosin is holding onto the actin and pulling each side closer together, making it much shorter. To help regulate a muscle contraction and prevent the myosin from grabbing hold any old time, actin will contain what's known as a tropomyosin and troponin complex, otherwise known as a tropocomplex. Tropomyosin will block the binding site on actin until it is time for a muscle contraction to occur. A motor neuron is necessary in order to activate a muscle contraction and stimulate the removal of that tropomyosin. Once stimulated, the motor neuron will send a signal down its axon in the form of an action potential, which will then cause the release of its neurotransmitter, acetylcholine. The acetylcholine is secreted into the synaptic cleft of the neuromuscular junction. There it will bind onto a receptor protein on the sarcolemma, which will then stimulate an action potential into the muscle fiber. Underneath the sarcolemma and surrounding each myofibril is a structure known as the Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum is responsible for storing calcium. Calcium will play a critical role in the contraction of a muscle. The action potential will move along the sarcolemma and down a transverse tubule, also known as a T-tubule, which will help to propagate that stimuli deep into the myofibrils below. The action potential will move into the sarcoplasmic reticulum, which will then stimulate the release of the stored calcium directly onto the myofibrils. As calcium is released from the sarcoplasmic reticulum, it will then bind onto the troponin component of the tropocomplex. This will cause the tropomyosin to shift and move away from the actin. This will expose myosin's binding site on actin. Now, myosin is going to need a little bit of energy in order to get up and interact with the actin.
the way ATP works is when it releases one of its phosphates, energy is released. This allows myosin to take on some of that energy and stand right on up. Dropping that phosphate, ATP will turn into ADP, giving it lots of energy to pick up that myosin and grab hold of that binding site. With one last bit of energy, ADP gives a good push, releasing from myosin. Myosin will then pull onto that actin, drawing both ends of that sarcomere close together, shortening our muscle fiber. Now ADP is completely depleted of all of its energy, so myosin will stay tightly held onto actin until more energy comes along, releasing it. So a new ATP will come along and tug that myosin head right off of that actin. This process will continue as long as there continues to be a nerve impulse and the presence of calcium. This whole process is known as the sliding filament theory of a muscle contraction, resulting in the shortening of a muscle. When there's no longer a nerve impulse, the calcium is pumped back into the sarcoplasmic reticulum. The tropocomplex will shift and go back over the actin, covering that myosin binding site, bringing everything back to rest. That is,